Vision. Vision is the ability to detect light patterns from the outside environment and interpret them into images. Animals are bombarded with sensory information, and the sheer volume of visual information can be problematic. Fortunately, the visual systems of species have evolved to attend to the most important stimuli. The importance of vision to humans is further substantiated by the fact that about 1/3 of the human cerebral cortex is dedicated to analyzing and perceiving visual information. Light. As with auditory stimuli, light travels in waves. The compression waves that compose sound must travel in a medium, a gas, a liquid, or a solid. In contrast, light is composed of electromagnetic waves and needs no medium. Light can travel in a vacuum. Figure 1. The behavior of light can be discussed in terms of the behavior of waves and also in terms of the behavior of the fundamental unit of light, a packet of electromagnetic radiation called a photon. A glance at the electromagnetic spectrum shows that visible light for humans is just a small slice of the entire spectrum, which includes radiation that we cannot see as light because it is below the frequency of visible red light and above the frequency of visible violet light. Certain variables are important when discussing perception of light. Wavelength, which varies inversely with frequency, manifests itself as hue. Light at the red end of the visible spectrum has longer wavelengths and is lower frequency, while light at the violet end has shorter wavelengths and is higher frequency. The wavelength of light is expressed in nanometers. nm. 1 nanometer is 1/1000th of a meter. Humans perceive light that ranges between approximately 380 nanometers and 740 nanometers. Some other animals, though, can detect wavelengths outside of the human range. For example, bees see near ultraviolet light in order to locate nectar guides on flowers. And some non-avian reptiles sense infrared light, heat that prey gives off. Wave amplitude is perceived as luminous intensity, or brightness. The standard unit of intensity of light is the candela, which is approximately the luminous intensity of a 1 common candle. Light waves travel 299,792 kilometers per second in a vacuum, and somewhat slower in various media such as air and water. And those waves arrive at the eye as long, red, medium, green, and short, blue waves. What is termed white light is light that is perceived as white by the human eye. This effect is produced by light that stimulates equally the color receptors in the human eye. The apparent color of an object is the color, or colors, that the object reflects. Thus a red object reflects the red wavelengths in mixed white light and absorbs all other wavelengths of light. Anatomy of the eye. The photoreceptive cells of the eye, where transduction of light to nervous impulses occurs, are located in the retina, shown in Figure 2, on the inner surface of the back of the eye. But light does not impinge on the retina unaltered. It passes through other layers that process it so that it can be interpreted by the retina. Figure 2b. The cornea, the front transparent layer of the eye, and the crystalline lens, a transparent convex structure behind the cornea. Both refract, bend light to focus the image on the retina. The iris, which is conspicuous as the colored part of the eye, is a circular muscular ring lying between the lens and cornea that regulates the amount of light entering the eye. In conditions of high ambient light, the iris contracts, reducing the size of the pupil at its center. In conditions of low light, the iris relaxes and the pupil enlarges. The main function of the lens is to focus light on the retina and fovea centralis. The lens is dynamic, focusing and refocusing light as the eye rests on near and far objects in the visual field. The lens is operated by muscles that stretch it flat or allow it to thicken, changing the focal length of light coming through it to focus it sharply on the retina. With age comes the loss of the flexibility of the lens, and a form of farsightedness called presbyopia results. Presbyopia occurs because the image focuses behind the retina. Presbyopia is a deficit similar to a different type of farsightedness called hyperopia, caused by an eyeball that is too short. For both defects, images in the distance are clear but images nearby are blurry. Myopia, nearsightedness, occurs when an eyeball is elongated and the image focus falls in front of the retina. In this case, images in the distance are blurry but images nearby are clear. There are two types of photoreceptors in the retina: rods and cones, named for their general appearance as illustrated in Figure 3. Rods are strongly photosensitive and are located in the outer edges of the retina. They detect dim light and are used primarily for peripheral and nighttime vision. Cones are weakly photosensitive and are located near the center of the retina. They respond to bright light, and their primary role is in daytime color vision. The fovea is the region in the center back of the eye that is responsible for acute vision. The fovea has a high density of cones. When you bring your gaze to an object to examine it intently in bright light, the eyes orient so that the object's image falls on the fovea. However, when looking at a star in the night sky or other object in dim light, the object can be better viewed by the peripheral vision because it is the rods at the edges of the retina, rather than the cones at the center, that operate better in low light. In humans, cones far outnumber rods in the fovea. Transduction of light. The rods and cones are the site of transduction of light to a neural signal. Both rods and cones contain photopigments. In vertebrates, the main photopigment, rhodopsin, has two main parts: Figure 4, and opsin, which is a membrane protein, in the form of a cluster of alpha helices that span the membrane, and retinal, a molecule that absorbs light. When light hits a photoreceptor, it causes a shape change in the retinal, altering its structure from a bent cis form of the molecule to its linear trans isomer. This isomerization of retinal activates the rhodopsin, starting a cascade of events that ends with the closing of Na+ channels in the membrane of the photoreceptor. Thus, unlike most other sensory neurons, which become depolarized by exposure to a stimulus, visual receptors become hyperpolarized and thus driven away from threshold. Figure 5. Trichromatic coding. There are three types of cones, with different photopsins, and they differ in the wavelength to which they are most responsive. As shown in Figure 6, some cones are maximally responsive to short light waves of 420 nanometers. So they are called S cones. S, for short. Others respond maximally to waves of 530 nanometers. M cones, for medium. A third group responds maximally to light of longer wavelengths, at 560 nanometers. L, or long, cones. With only one type of cone, color vision would not be possible, and a two-cone, dichromatic, system has limitations. Primates use a three-cone, trichromatic, system, resulting in full color vision. The color we perceive is a result of the ratio of activity of our three types of cones. The colors of the visual spectrum, running from long wavelength light to short, are red, 700 nanometers, orange, 600 nanometers, yellow, 565 nanometers, green, 497 nanometers, blue, 470 nanometers, indigo, 450 nanometers, and violet, 425 nanometers. Humans have very sensitive perception of color and can distinguish about 500 levels of brightness, 200 different hues, and 20 steps of saturation, or about 2 million distinct colors. Retinal processing. Visual signals leave the cones and rods, travel to the bipolar cells, and then to ganglion cells. A large degree of processing of visual information occurs in the retina itself. Before visual information is sent to the brain, photoreceptors in the retina continuously undergo tonic activity. That is, they are always slightly active even when not stimulated by light. In neurons that exhibit tonic activity, the absence of stimuli maintains a firing rate at a baseline, while some stimuli increase firing rate from the baseline, and other stimuli decrease firing rate. In the absence of light, the bipolar neurons that connect rods and cones to ganglion cells are continuously and actively inhibited by the rods and cones. Exposure of the retina to light hyperpolarizes the rods and cones and removes their inhibition of bipolar cells. The now active bipolar cells in turn stimulate the ganglion cells, which send action potentials along their axons, which leave the eye as the optic nerve. Thus, the visual system relies on change in retinal activity, rather than the absence or presence of activity, to encode visual signals for the brain. Sometimes horizontal cells carry signals from one rod or cone to other photoreceptors and to several bipolar cells. When a rod or cone stimulates a horizontal cell, the horizontal cell inhibits more distant photoreceptors and bipolar cells, creating lateral inhibition. This inhibition sharpens edges and enhances contrast in the images by making regions receiving light appear lighter and dark surroundings appear darker. Amacrine cells can distribute information from one bipolar cell to many ganglion cells. You can demonstrate this using an easy demonstration to trick your retina and brain about the colors you are observing in your visual field. Look fixedly at figure 7 for about 45 seconds. Then quickly shift your gaze to a sheet of blank white paper or a white wall. You should see an afterimage of the Norwegian flag in its correct colors. At this point, close your eyes for a moment. Then reopen them, looking again at the white paper or wall. The afterimage of the flag should continue to appear as red, white, and blue. What causes this? According to an explanation called opponent process theory, as you gazed fixedly at the green, black, and yellow flag, your retinal ganglion cells that respond positively to green, black, and yellow increased their firing dramatically. When you shifted your gaze to the neutral white ground, these ganglion cells abruptly decreased their activity and the brain interpreted this abrupt downshift as if the ganglion cells were responding now to their opponent colors, red, white, and blue, respectively, in the visual field. Once the ganglion cells return to their baseline activity state, the false perception of color will disappear. Higher processing. The myelinated axons of ganglion cells make up the optic nerves. Within the nerves, different axons carry different qualities of the visual signal. Some axons constitute the magnocellular, big cell, pathway, which carries information about form, movement, depth, and differences in brightness. Other axons constitute the parvocellular, small cell, pathway, which carries information on color and fine detail. Some visual information projects directly back into the brain, while other information crosses to the opposite side of the brain. This crossing of optical pathways produces the distinctive optic chiasma, Greek for crossing, found at the base of the brain and allows us to coordinate information from both eyes. Once in the brain, visual information is processed in several places, and its roots reflect the complexity and importance of visual information to humans and other animals. One route takes the signals to the thalamus, which serves as the routing station for all incoming sensory impulses except olfaction. In the thalamus, the magnocellular and parvocellular distinctions remain intact, and there are different layers of the thalamus dedicated to each. When visual signals leave the thalamus, they travel to the primary visual cortex at the rear of the brain. From the visual cortex, The visual signals travel in two directions. One stream that projects to the parietal lobe, in the side of the brain, carries magnocellular, where, information. A second stream projects to the temporal lobe and carries both magnocellular, where, and parvocellular, what, information. Another important visual route is a pathway from the retina to the superior colliculus in the midbrain, where eye movements are coordinated and integrated with auditory information. Finally, there is the pathway from the retina to the suprachiasmatic nucleus, SCN, of the hypothalamus. The SCN is a cluster of cells that is considered to be the body's internal clock, which controls our circadian, day-long cycle. The SCN sends information to the pineal gland, which is important in sleep, wake patterns and annual cycles. Section summary. Vision is the only photoresponsive sense. Visible light travels in waves and is a very small slice of the electromagnetic radiation spectrum. Light waves differ based on their frequency. Wavelength equals hue, and amplitude. Intensity equals brightness. In the vertebrate retina, there are two types of light receptors: photoreceptors, cones and rods. Cones, which are the source of color vision, exist in three forms: L, M, and S, and they are differentially sensitive to different wavelengths. Cones are located in the retina, along with the dim light, achromatic receptors. Rods. Cones are found in the fovea, the central region of the retina, whereas rods are found in the peripheral regions of the retina. Visual signals travel from the eye over the axons of retinal ganglion cells, which make up the optic nerves. Ganglion cells come in several versions. Some ganglion cell axons carry information on form, movement, depth, and brightness, while other axons carry information on color and fine detail. Visual information is sent to the superior colliculi in the midbrain, where coordination of eye movements and integration of auditory information takes place. Visual information is also sent to the suprachiasmatic nucleus, SCN, of the hypothalamus, which plays a role in the circadian cycle. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.